Structure and function of living things is going to be the title of Unit 3. We are going to look at living things and all the things that make up living things and the things that are needed for them to support life. Cell theory is the idea of all living things are composed of cells. Okay, that is the first part of the cell theory. If you think about, an amoeba or a protist, they are unicellular organisms, so they are one-celled, but they are still composed of a cell. If you think of things as large as whale or elephants, they're multicellular organisms, but they're still composed of thousands and trillions of cells. Same thing with humans. I think we're up to 10 trillion cells in one human. And we continue to shed and regrow those cells every day. So part 1 of cell theory, all living things are composed of cells. The second part of cell theory is cells are the smallest unit of life. Okay, they're the basic structure. We cannot take a cell apart and then expect the parts of the cell to sustain life. It is the whole cell. We need every part of the cell to help support life in an organism. The third part of the cell theory is that cells come from pre-existing cells and cannot be created from non-living material. This is to say that living things do not spontaneously exist. We don't get flowers from the soil. We get flowers from the seed of other flowers that are buried in the soil. Okay, living things come from pre-existing cells, and we'll talk a little bit about how 2 scientists did experiments to test this out. Cell theory was really pushed and supported with the development of the microscope. Okay, before this we would only see things that we could see with the naked eye, or maybe a small crafted hand lens, but we couldn't get the magnification that we can with the development of the microscope. The evidence for this, so a couple of scientists that have really pushed for cell theory and to support all 3 parts of it are, the first one is Robert Hooke. He was the first to coin or use the term cell. Before this, it went that a cell was a small room that a monk lived in, very similar to kind of like a jail cell. It's a small room that has the basic necessities for life, and that's about it. Hooke also used a microscope to examine cork, which is the inside of a bark before you get to the wood of the tree. Anton van Leeuwenhoek is officially the discoverer of cells. He gets that title. He also is sometimes called the father of biology. He observed the first single-celled organism, scooting across the slide of the microscope. And then we also have Pasteur's experiment. Louis Pasteur we talk about with bacteria a lot because we talk about pasteurizing milk and get rid of bacteria that way. What he did to help disprove spontaneous growth of life is he kind of copied Thomas Reddy's experiment with meat, only he did it with fancy glasses, and we'll show you guys that in the next slide. And then lastly, Robert Remek viewed cellular division under the microscope, so he was being able to see cells come from other cells, which really support the principle 3 in cell theory that living things come from other living things. So here's a slide of Reddy and Pasteur's experiment, and you can see that Reddy put meat in 3 jars and then covered them in different ways to see what would be, what would turn out. So in the first one, he covered with paper, and the second one he left open, and the third he put gauze on top. What he did is leave this out for a couple of days, and then he came back and looked at it. So with the one with the paper on top, you can see that there are no flies in it due to the paper. So the flies weren't able to land on the meat, lay eggs, and then develop into larvae or maggots. The open one, you can see that there are flies. The flies come and go. There's not necessarily maggots because they were able to eat the meat and develop into flies. The last one is you can see that there's no flies in there, but they are outside, and then especially on top there are maggots. So this allowed the flies to, lay eggs on the top and then fly away. So what left, but the maggots weren't able to eat the meat and grow to develop flies, so that's why they're still on top. Pasteur did the same thing, only used fancy swan neck flasks. He heated broth in both of them to destroy any bacteria. The one, he left the nice swan flask or swan neck on, which doesn't allow air and bacteria to get up in there. The second one he broke off, which allowed air and bacteria to get in, and then after a while you could see that there was growth in the flask.